Good afternoon, everyone. Thank you for being on time. Let's start our program of the afternoon of the last day of this symposium. We are now going to have a lecture of Dr. Jor Lindenau from Zeiss. I'm sorry. And he is originally a biologist, and he specialized in neurobiology during his PhD. And from 2000, he joined Zeiss, where his task was first to establish application for laser scanning confocal microscopy. And during the following years, he also works as an imaging specialist, providing worldwide application support for local Zeiss organizations. I had the opportunity to have an online meeting with him. He made some demonstrations, some work for our group. And from 2012 and 2014, he also managed the Zeiss microscopy distributor network in the Middle East region. But today, we're talking more about science, so I don't want to lose any minute of his presentation. Thank you very much.
Thank you very much for the nice introduction and for the opportunity to talk today. What I would like to do, I would like to lower the resolution from the previous talk, but increase the volume of the imaged area. So which means I would like to talk about scanning electron microscopy volume imaging. And as you all know, there are 3 different major techniques how to approach a volume. And I would like to give a short update on each of the technologies first before I lower the temperature and talk about cryo. So the first one here is array tomography, and I think it is all well known. I just want to give one example as an update. So the technique where you acquire serial sections with the automated tape collecting ultramicrotome is used widely in order for brain connectomics. And what you do here, you collect the samples on a tape, glue the individual sections on a tape, and then you analyze one tape after the other. And you can imagine if you do this with a single beam system and want to approach a cubic millimeter, that takes quite a while. It was calculated that for this cubic millimeter here, the pure scan time without any image analysis would be about 6 years of scanning. So, and with the multi-beam system here in Jeff Lichtman's lab, it was done in 4 months for the first cubic millimeter ever published, and this is out now since almost now 2 years, and everybody can access these nice data set on the petascale level, and the resolution is enough to look down to the individual synapses. So this is more on the image analysis side, and what we have added to the field of array tomography in the recent past is a specialized detector which has special imaging capabilities under low kV, for low kV imaging. So this is for single beam systems, the other one was just the multi-beam. But despite from this, there's not much new on our side, and just there is the new detector allows to scan faster and increase scanning time for single beam systems in array tomography. So the second technology here is obviously serial block phase imaging, and there is indeed something new from our side, because after waiting quite a while, finally we came out with our Zeiss own serial block phase device, and this is based on the original Denkotome technology or design. So it is a door-mounted system. We redesigned it completely, making it very rigid and stable. As you see here inside, this is how it looks like in our days. This is the sample holder, which goes down here. Here we see the diamond knife. Okay, sample thickness, what we can cut is 25 nanometers. then it's a completely automated system. And also here there is a special detector, a new one for low volt, also low kV imaging, so in a range between 1 and 2 kV. This is typically what we use for these kind of samples. And of course it comes with focal charge compensation, which becomes essential if you have samples where there is empty areas of resin, because those kind of samples are prone to charging, and these local charge compensation, this little push of nitrogen gas here, which can then be ionized, takes the charging away and increases the contrast of the imaging quite a lot. This is very essential. For instance, if we look at tissues like we have seen on the first talk of Professor Ochs here, if you look in the lung, so there is many areas where there is empty resin, and for this type of sample, focal charge compensation is absolutely necessary in order to get high contrast images here. Okay, so as I said, just a short update on this, and I would like to spend a little bit more on crossbeam imaging, starting with well-known basics, but let's have a look at the sample size, what we can approach with this kind of technology. So with the typical approach where we have a sample embedded in a block of resin, what we can do with this is doing the certain measures here, just imprint this kind of fiducials on the top of the sample, then cover it with a carbon layer to increase the contrast, then drill or dig a trench here, polish the trench, and then start imaging. So this is what has been done all the time already, and the typical size of the sample, what you can approach here is, let's say, a single cell. The Z resolution which can be achieved is by usage of this pattern here for the guidance and for the measurement of the forward movement in Z is about 3 nanometer, between 3 and 5. This is what we can achieve. And this can be used for single cells like this, or cellular organelles, up to the level of cells, as we see here. And but if we want to get images, 3D representation of objects larger than a cell, so then there's a challenge due to the following phenomenon. First of all, as we have heard already, the information here gets darker if we go in, so we lose information here, electrons. And the second is redeposition as a material sample. If you just try to cut out these lamella, so we see a redeposition here at a certain depth. So that's a problem. And this allows us, or this stops us on to look at larger samples in this case. So and there was a nice idea. It comes, I think, from the EMBL here. And it's called fish kebab. And what they did, they put these large samples here on little sticks, so that's why it's a fish kebab, and let all the resin drop off before it finally polymerizes. And by doing this, afterwards they can just glue the sample on a substrate and then start milling into this animal, so to say, and even take images from the complete animal here during a long time with a very stable image acquisition. This was done the first time, I think it's a couple of years already ago. The complete zebrafish larvae here, which was 80 microns long, was imaged by cutting 10,000 sections with a 5 nanometer axial resolution, this kind of volume. So, and then for a long time, these kind of data just were available, and this is just raw data without any segmentation, whatever, just the gray intensities here. And now we see the first publications where these data actually have been used in order to reconstruct the nervous system of the complete elegans here. Okay, and so this is based on exactly the raw data which has been acquired for this very large volume. And so what to do if the volume becomes even bigger? So if you even want to look at larger volumes. So what we have to do then, and this is, this is a collaboration with the Francis Crick Institute, with Carlos Bosch here. They have a sample have the bulbus olfactorius of a mouse actually, and they investigate some nervous circuits here, and they start with a label, with the in vivo labels to photon, after they sacrifice the mouse and preparation of the bulbus olfactorius. They overlay these two, and this one, what you see here, this is an overview scan of the complete organ with an X-ray microscope. So what we do now have in the second step here is X-ray microscopy plus 2 photon, and this helps us to identify the area which we would like to look at higher resolution. So what we use now is we use a special preparation device, a laser, in order to cut this material free. This was the very first approach here we did. And then later on, so we got this pillar here, and then the pillar in this case was used for synchrotron tomography. And this is important because the synchrotron, they don't like edges. So they really prefer round samples without any edges to prevent artifacts when this is rotated. But nevertheless, and by the way, here you see the laser attached to the crossbeam system. So it was a little bit unusual that we took the sample inside the crossbeam, took it out again, and then they brought it to the synchrotron. Nevertheless, the same workflow is also possible by creating not only one pillar, but a number of pillars, and this brings us to the problem that if you have a very valuable sample where the different areas of the sample represent different functionality, so in this case you don't want to lose any material of your sample, and so this very high slope angle, what the laser allows here, leads to the fact that we don't lose a lot of material, and we can just take different samples from the same initial probe in order to be very efficient in sample preparation. What we had to do, because then when we did this the first time, then we realized that, okay, we can break these pillars off and put it in the crossbeam system, but then we don't know which one was where, so we had to start a labeling with the laser on the top of the pillars to recognize which one belongs to which area. And then you can just perform normal FIP milling on this, and you don't have to cut material away which you're not interested in this case. Yeah? So this is a very straightforward and very efficient workflow. Okay. So there is a lot of software, of course, in the systems. I think this is quite normal everywhere. So we have, there is some image processing software, there is software for object classification, deep learning stuff. We acquired the company Arrivis, which is now fully integrated, and then software for visualization as well. And with this, I would like to lower the temperature a little bit and come to the second part of the presentation. Where we stay with crossbeam imaging, but under cryo conditions. This is one of the very first images acquired in the crossbeam without, this is just the raw data, no destriping, no denoising, nothing. This is from, okay, quite some years ago already. And so we see contrast without any labeling, so we know this all. And the workflow which we would like to concentrate in today here is potentially there are two different methods of sample preparation. We have seen a lot of plunge freezing. I would like to focus a little bit on high pressure freezing here. And then what we do, we use the best microscope with the highest potential resolution. So area scan confocal in an upright configuration with a dipping lens here. And then we acquire the fluorescence, overlay this with the cryo data, and then you have two different options, either to go for pure volume imaging or create a lamella and move to a cryo-TEM. The workflow, which okay, and this is how this equipment looks like. So we have a central mounting unit here. This is from Quorum, and then we have a Lincoln stage mounted under the confocal microscope, and the Quorum stage, the loading stage for the crossbeam system here. This is how this loading stage looks like. So we have a sample holder. which can accept naked grids, clipped grids, or planchets. And then there's adapter for the linking stage and for the crossbeam. So in the first place, the sample goes in the cryo confocal microscope, which can be used for wide field imaging, of course, but also for confocal. And for confocal, as we all know, it is very challenging not to warm up the sample, so we would like to avoid any kind of devitrification, which does start already at minus 140 degrees. So, and what we take care of here is to use all the light which comes back from the sample by using an array detector to detect the fluorescence light. And the array detector is good for sensitivity, but for higher resolution on the other hand side as well. Nevertheless, we need to keep in mind that we always have a distance between the sample and the objective lens. So the NA is kind of not the same as you have in the dipping lens. So, and from this point, one more important thing is that the the confocal microscope also allows to set up a reflection channel which shows the ice surface. So we always know how deep under the ice the fluorescence is. It's quite a nice additional feature what we can use here. And then we go into the crossbeam system and acquire volume images in the crossbeam system. And I would like to show one application example we developed together with Professor Nico Samadijk from Radboud University in the Netherlands. He works on fish scales, and he is interested in extracellular matrix deposition of calcium. And the workflow, what they did here is they do live cell imaging on the zebrafish fish scale, then they freeze it, then they go back to the confocal microscope, do the same imaging from the same fluorescence labels with under cryo conditions, go to the volume imaging in the cryo crossbeam, cut a lamella, lift lamella out and go to cryo TEM. So they use an objective lens for the observation. This is the holder of the high pressure freezer from cryocapsule in this case, and if they press a button, then after 1.5 seconds the sample is frozen. So they have the time resolution of this workflow is about 1 to 2 seconds, and then it goes into the cryofluorescence microscope. So, and then one important thing here is that of course you need to have kind of a reference contrast between the cryo imaging in the fluorescence microscope and in the crossbeam later. And there was a nice innovation by Anna Takivat here. So she created a finder top, and by freezing, high pressure freezing, you see the imprint of the finder top on the ice, and this can be seen in the reflection channel of the fluorescence microscope, but of course also in the surface image of the crossbeam. So this is then used in order to be applied for the correlation. So we see here, this is the confocal imaging of the frozen sample. We see the surface of the ice, this is the reflection channel, with the information in X and Y about the position of the region of interest, and then there's two different layers of bone osteoblast forming layer and other cells here, and we also see mitochondria. The sample is transferred into the crossbeam system by overlaying the two different images from the confocal system. Here, this is the region of interest in the confocal data set. Then we tilt it. This is the crossbeam image. And so we identify the region of interest by the fluorescence. This is the defined milling trench. Here you see, this is the kind of data we would like to acquire. This is the trench in front of this area to mill down. And of course, we always know the depths of the region of interest by the information from the confocal microscope. And then this is the result of approaching the end of the lamella. So we see different layer of collagen organization here. And if you look deeper into this, we can even realize that on the mitochondrial level, so we see the mitochondria here, and we can identify these exactly with the fluorescence data. So we know exactly where we are on a subcellular level. And this is now coming to higher resolution. So here we start with a voxel size of, this should be a movie actually. We start with a voxel size of 20 by 20 by 40. nanometer in the crossbeam, and this is the area of interest where the collagen is obviously organized, or the calcium calcification, and then it goes to higher resolution, 10 by 10 by 20, and the highest resolution what they achieved here was a 5 nanometer volume, pixel volume, with a little shift, but anyway there was something like 1,600 slices acquired with this resolution, and you can see the individual collagen fibrils here of a size of about 60 nanometer, and you can also see a different orientation in this special area here. So then the last step here was, of course, after approaching an area of interest, then there was a lamella. I have to speed up a little bit. Then there was a lamella cut free, and in this case we still used tweezers. Now we prefer to use a probe and solder it to the probe. It's much more convenient. And then the lamella was mounted to an SEM to a half moon grid, and I'm not allowed to show the data because they have to be published. But they are there, so it's about orientation of collagen fibers and fibrils. But the nice thing here is that there is Nico put this movie together where we have all these complete multi-scale imaging from the complete organ down to almost sub-nanometer resolution. So this is the high-resolution fluorescence what we see now. This is the finder grid as a bridge channel for the contrast. This is the same target area by cryo fluorescence, also fluorescence under cryo condition. And then this was approached for, as we said before, to FIB-SEM volume imaging. This is the trench. This is again the area. This is the trench. This is the volume which was imaged by FIB-SEM. And after this volume, so now we swap the view. We look from the EM imaging direction now to this, to see the side view. Exactly. So we see the different fluorescence layers. When milling into the sample, and so now we overlay this data with the crossbeam information in this area here. So this is now the different fibrils in progressing towards that direction, and then it ends at the position where the lamella was created and taken out of the sample. Okay. Yeah, so now it's continuing a little bit further, and then we know exactly in which cellular and tissue context these lamella was created, and then in the lamella there's two different sites for tomograms. So they created two tomograms of this lamella, one here and one further down, in different layers where you don't know exactly which cellular layer they are, and then they can refer this with the TM data. And that's it actually.
Thank you very much. And we are just in time, but if someone has questions, can make one question or talk to Dr. Lindenau during the coffee break. And for those who will be in Olinda, he will be there too, because so many new acquisitions for presenting and researching. Yeah.
One question. One row.
Bruno.
May I still? Actually, the first is maybe not so important. You said that you know the depth because of your confocal imaging, but what is the set resolution actually in AIRIScan?
So, I mean, the Z resolution depends on the NA of the objective lens. So,
I mean the one you can use in cryo.
So in cryo we have 0.9, so we might be somewhere at half a micron in Z.
Okay.
Yeah.
Thank you. And the next one is how do you say, how deep in that direction can you take slice and view images before you're at the end of the beam shift?
I don't know. I would have to ask my my technicians to
Okay.
answer this. So I'm sorry. Yeah.
Okay,
But,
thank you. But
obviously, with the okay, you're asking when do I have to reset the beam shift? Yeah.
Right.
But we did it we did it successfully with the CE elegans for 80 microns. So, but resetting and finding the same position again.
18 microns.
80.
Oh,
80.
80.
80
Okay.
microns.
Yeah, yeah. Anyhow, actually, I wanted to start with congratulations to size, because you really got a big step forward from what I have seen last time.
Okay, that's good. Thank you.
Thank you. So let's move to the next part of our symposium with oral presentations, short talks, and we're starting with Professor Kilder Miranda, that is very well known for most of you. He is a director of the microscopy unit at Xenabio. He is a professor here at the Instituto de Biofísica. He's a former president of the Brazilian Society for Microscopy and also for the Inter-American Society of Electron Microscopy Societies. And, well, I will not lose any more time because if so, I will stole his time of presentation. Thank you, Kilder. And he will talk about the identification of spatially resolved ion nanodomains in subcellular compartments of Trypanosoma cruzi.
Thank you, Marcia. So I was overwhelmed with so many things to do. I wasn't sure if I was giving a talk. Then I decided to drop a message to Carol. Said, Carol, would you give me some time? And the reason that I did that is that I'm not going to talk about cryo-EM. I'll just show one slide with cryo-EM that we did with Rodrigo. But this is something that I really cherished in our field. I think that there's a lot of potential to explore the IO identification capabilities of the systems that we are using. So our models here, so as you know, we work with parasites, protozoans, and some cases some nematodes. And these parasites, they have very interesting processes and structures that are used to govern these processes that are very interesting, both from the cell biology point of view, and also as they don't exist in mammalian cells, they are also important targets for development of new drugs, new antiparasitic drugs. And among these processes and structures, so we are mainly interested in transporters from ions and osmolytes. I'm going to talk about only the trypanosome, not with the work that we have with malaria. And in the trypanosome, these transporters and ions and osmolytes are, we have two main structures that we study, the acetylcarboxysomes and the contractile vacuole complex. So a brief introduction in the acetylcarboxysome. So it's basically, I won't have time to go through all the ions, transporters and pumps. Basically, it's a bag full of ions that was seen actually since the first observations of Trypanosoma cruzi. This is a drawing from the Carlos Chagas, who described the disease and the parasite also, and the life cycle and the vector. So he did everything. And in his drawings, we could already see these dots here, which represent the volutin grains that we know now that correspond to the acetylcarboxysomes. So if we look at the electron microscope, these cells are thin enough to be seen as a whole mount in the microscope. So Grant was mentioning about whole cells, right? So, but if we use an energy filter and tune the contrast, instead of looking only for the zero loss imaging, if we tune the contrast, well, we can improve a lot both contrast and also chromatic aberration. So with that, we can see very clearly these dots inside the cells. And this was the work that we did where we showed that all the trypanosomes, malaria parasites, toxoplasma, all of them contain acidocalcisomes, which are these black dots. If we combine these techniques with X-ray microanalysis, so in this case we did STEM, energy filtered STEM, so we used energy filter just to do contrast tuning and collected, as the samples are too thick to do to collect ion elemental signals, right? They're too thick. We use the X-ray detectors to get the elemental composition. So there we can see phosphorus. Marcia, could you give me a pointer? This one's not. It's not working. Okay. And so then here we can see phosphorus, magnesium, calcium, potassium. So with this work, we showed that they are highly dynamic. It is on. Not working. So. Okay, thank you. Okay, thank you. Now it's okay. Thanks. So then you see that they are not just trash for ions, they are really dynamic. If we change the composition of the medium, if you stimulate the cells, they can change the composition, change the shape, change the physiology. I won't have time to talk about this. We did almost 20 years ago, right? And then the reason that we do that is because these parasites, they have a very complex life cycle, and the parasite faces several challenges during the life cycle. So they are in different tissues, in different environments. And one of the challenges is the osmotic challenge. For instance, when the parasite is in the feces of the urine feces from the insect vector, and they invade the whole cell, they were adapted to 1,000 milliosmoles osmotic pressure, and when they invade, they face 300 milliosmoles per kilo pressure, osmotic pressure, which is isosmotic condition for most of the cells, but for a cell that was adapted to 1,000, this represents a hyposmotic shock, and the parasite has to deal with that, and it deals with that through the contractile vacuole complex, right? And so. And what we do in our group here, we study the biochemical pathway, the signaling pathway that controls the osmoregulatory system in T. cruzi. So I won't have time to talk about all these proteins, but what we do, we produce mutants that are either very efficient or are not efficient to osmotic regulation. So for instance, here these are wild type cells, and these are cells that are super efficient, submitted to the same osmotic pressure at the same time. See what happens, they swell, but then they slowly recover. So then we are using this as a model. As we have wild type cells, we have cells that overexpress enzymes that make them very efficient, and on the other side they are not efficient at all, and then we are comparing the structure of the contractile vacuole and acidocalcisomes. Ingrid has done a lot of work with the contractile vacuole and the link with the flagella pocket, both with cryo-EM and free substituted samples. I won't have time to talk about it. I will focus on this interaction between acidocalcisomes and contractile vacuole. The idea here is that the acidocalcisomes, they contain a lot of ions, and once they interact with the contractile vacuole and fuse, they release the ion content within the contractile vacuole lumen, increasing the osmotic pressure towards the contractile vacuole. And we were very interested in what was happening in this process here. We were searching to see if we could catch the transfer of ions between these two compartments. We could see it through tomography. We did a lot of tomography to see this fusion process, but we wanted to see the transference of ions between these two organelles. And one main point that we have when we do high pressure freeze and freeze substitution, or we do routine works that we lose all the ions. This is well known in analytical, biological analytical EM, and we lose part of the ion content within the acetylcalcosome. So what we did, this was work done by Wendell. What we did was to adhere cells to grids and plunge freeze these cells and freeze dry the cells, right? And these are the results that we got with this type of samples, so this type of sample preparation. So freeze dried cells, we can retain the ions, we can map them. And what we started to see here when we do some line scans, that the ions within the acetylcalcosomes using new instruments, so these now are new instruments, not those that we used like 20 years ago that we had to sleep in the lab waiting for the maps, right? Manually adjusting drifts. This was done with a modern instrument. We saw that the ions seemed that were not co-localizing the matrix of the acetylcalcosomes. So what we did was to use an instrument that has 4 integrated EDS detectors, and this is very important because if we want to do tomography, combine stem tomography with ES tomography, we have a problem when we talk about microanalysis, because if you use a regular instrument with only 1 detector, which is the case of most of the analytical TEMs. When you tilt your samples towards the detector, it's fine, you get your signal, but when you tilt to the other side, you lose the signal completely. So using this type of configuration, we're able to get elemental tomograms from whole cells. So these are the stem images. These are, for instance, phosphorus mapping that we did along the tilt series, and this is phosphorus, magnesium, calcium, zinc. So this was elemental distribution within the acetylcalcosomes. And for our surprise, when we reconstructed this data, we saw that the elements inside the acetylcalcosomes are not really a homogeneous matrix, where we saw the formation of nanodomains inside the acetylcalcosomes. And the cations, the Nivelian cations there, they almost exclude themselves. So here we see the phosphorus map, magnesium, see how magnesium, then we see calcium in red, and see how they just fit, they exclude themselves. So we saw the formation of nanodomains inside the acetylcalcosomes. This was something that was very cool, because we went to see our old elemental maps and saw that it looks like in 2D images that they were really organized in nanodomains. And if you take images from different organelles in the same cell, we see how they are really different. So this might represent different physiological states. So we are really excited about these results. And when we went to the literature, we did some measures, so correlation coefficient, just to show that they were really not localized. For instance, when you have phosphorus and phosphorus here, we have number 1, which means full colocalization, and we can see that the different elements, they do not colocalize in the matrix of the organelle. And when we look at the literature, this is actually not new. So all the theories that place ions within different volumes, different compartments, they don't apply if you are talking about a nanoscale compartment. So there, it's not the simple diffusion theory that we apply for solutions that we prepare. This doesn't apply for compartments with nanoscale dimensions, because you have a lot of membrane potential, things that happen there, for instance, electron diffusion. So what we saw is that, in theory, what we expect is not to see a homogeneous distribution of ions, but actually ions as clusters within the organelle. So we tried to understand that. We did some molecular modeling. We're organizing polyphosphate chains inside simulating polyphosphate chains inside the acetylcalcosome. So the phosphorus that we're seeing is organized mainly as a polyphosphate chain there. So polyphosphates bound to monovalent cation, sodium, and magnesium. What we saw is that when we have an extra positive charge here, it links two different polyphosphate units. So this explains why we have more linear distribution when we have polyphosphate bound to sodium or polyphosphate, and more aggregate when we have polyphosphate bound to magnesium. So this is more or less what we were seeing in our data. But I'm really excited about this field because novel systems are on the way. So this is a recent work from Nestor Zaluzek, where he is developing, together with Thermo Fisher, these racetrack-style silicon drifter detectors. So they are really improving a lot the sensitivity of the system. So in some cases you can have like this circumferential detection capability, and this would improve a lot our detection sensitivity. And I think that if we're able to combine it with cryo, we'll be able to get elemental information from frozen samples. And if we consider that a lot of the drugs that we use to treat these parasites or for other purposes now, there's tons of works that they combine different, somebody's calling me. That they combine different elements with different drugs, so we could even map these drugs inside cells. So this is something that is really exciting in the field. So if we look at our data, so this is a data that Ingrid got at Cenepain with whole cells. This is a section from a virtual section from a tomogram in frozen cells. What we see actually is that sometimes the electron dense core of the acetylcalcisomes, they look organized more like this, and sometimes they are fragmented and looking like this. So maybe we could correlate these two data. This is our dream, to have frozen samples and be able to correlate analytical and anatomical data from the cells. So this is really exciting. So I have to thank Wendell that did most of this part of the work. I think that Wendell is here somewhere. I don't know where. Yeah, he's at the back. And Ingrid that did a lot with cryo work. Camila, which works with Plasmodium, but was always working together with us in the cryo work. And Vanya and Adelia, they are invisible. They are just always backing up us a lot, and they don't want to appear in our work, but I would like to thank them. So Vanderlei, you know, Pedro Pascucci and Tassio did the molecular modeling, and Glenda is a postdoc that is working in Manaus. So we are really excited with the work that we are doing with Plasmodium vivax, and we expect to apply these technologies also to Plasmodium vivax. So our collaborators at the University of Georgia, University of Buenos Aires and California, and also our sponsors. And Masi, if you indulge me with just 30 seconds, I just wanted to mention that next week we're having our microscopy congress. For those that will be there, we are organizing a workshop which is dedicated to the career path for the imaging scientists. So this is for companies, for academics, for technicians, technologists, and we expect to have you there. And for those that won't be there, we will try to organize a virtual session so that you can also participate. So thank you very much.
Thank you very much. You did not explode your time. You were perfectly on time. Any questions? We can have one question from the audience. Otherwise, we can follow to the next presentation. Okay, Adam.
So in the world of fluorescence microscopy, there is something that is coming more and more, which is the hyperspectral analysis using phasor plots to segmentate information from different colors. I wonder if this can apply to segmentate elemental composition in EM using phasor plots.
Actually, I don't know if this will actually apply, because we have different signals. Actually, it's not really hard to segment it, the analytical data, because we only have the data from each element there. So it's like segmenting fluorescent signal, green signal, right? I think it's something that it's not really difficult to do because we have only one signal there. John.
Once you've shown these interesting nanodomains and these sort of alpha, if you go back to your own free structure images, can you see them by that too?
This is something that is very hard with free structure, because we don't have a good marker, right? We are not really sure if we're seeing acidocalcisomes, but I can tell you that when we began this work with different parasites, we had one plant trypanosome, Phytomonas. The matrix of the acidocalcisomes had some concentric rings, and this was easy to spot in free structure images, and this we could see. But with with T. cruzi, we don't know if we're looking at an acidocalcisome or other vesicles.
Okay, thank you. Our next speaker is Dr. Noemi Uaman from Centro Brasileiro de Pesquisas Físicas. She is going to load her presentation to the computer. Thank you. While she does it, I'll announce the title of her presentation: Effect of Aspartic Acid on Calcium Carbonate Crystallization, Nanostructural Analysis by Cryo-TEM.
Good afternoon. In this opportunity, I was be presenting work on the topic how calcium carbonate form. My name is Raquel Checa. I am postdoctoral fellow, doctoral research fellow at the Brazilian Center for Physics Research under the supervision of Professor André Rossi. In this presentation, article effect of aspartic acid of calcium carbonate crystallization in nanostructural analysis by Cryo-TEM. I want to start by imagine by and imagine abiotic calcium carbonate, which is a type of calcium carbonate formed during a synthetic synthesis. I imagine abiotic calcium carbonate which influence of produced by biological organisms. These crystals exhibit different mechanics for their crystallization. This is extremes of the great equation. The calcium carbonate exhibit three polymorphs with different morphologies: hexagonal battery, orthorhombic aragonite, and rhomboedral calcite. Currently, it is known that a precursor phase of crystalline calcium carbonate is amorphous calcium carbonate, which is closely involving in the nucleation and grown in this material. Amorphous calcium carbonate can form both abiotical and biotical. Abiotical amorphous calcium carbonate is high unstable in 1 to 2 minutes, and biotic amorphous calcium carbonate can last for months because it is produced in a biological environment. I question imports of calcium carbonate, how they are formed and transformed. This is essential for the compression of processes such as biomineralization, ocean acidification, the carbon cycle, etc. We know that occur through the classical pathway with crystal grown from cluster, or the no classic pathway involving amorphous precursors that transform in crystalline carbonates. Here, I'm showing diagram the multiple pathway of calcium production. This diagram is based of laboratory research of Professor Juan Diego Rodríguez Branco. Diagram showing the path of crystallization from a supersaturated solution of calcium carbonate going from lower to high supersaturation to final product calcite with the different size, form, shape, and surface properties. Calcium carbonate can form through many different pathways in mechanics. For example, they can be produced directly for the calcium carbonate solution or through the amorphous phases and sometimes through different metastable polyforms do carbonato calcio through mechanism of dissolution, recrystallization, or solid state transformation. In this work, we used a simple method, direct synthesis, where the source V, sodium carbonate, 1 molar, is placed in the source B, source A, calcium chloride dihydrate, two additive ácido aspartico was placed in source A, only for synthesis V. To analyze the dried sample, the synthesis were stopping after this 10 minutes and quickly filter ideas of 50 degrees centigrade overnight. The result the samples without additive show that zero-cheese additive pattern with their respective readable refinement using the pseudo-void per field 15. The readable refinement of this data indicates to spurious, no spurious phase, when 100% doctrinal phase show the typical FET year spectra of the calcium carbonate sample. The más intensas bandas are asignadas to calcite. Imagine C shown thermogravimetric analysis curve of calcium carbonate. The TGA revela that decomposition occur in 3 steps. Imagine D, E, F, same imagine showing the morphology of calcium carbonate nanostructures like layer of rhomboedral particle. Well, the sample with additives, GCRD, GCRD pattern synthetic using ácido aspartico is shown is respective refinement, refinement. In this synthesis, battery represent the main phase with KT 85% is the calcite phase with 15%. Show the FET respect of the calcite batterita band. Imagine a scene showing the micrometric spherical nanostructure. These nanostructures are composed of nanoparticles diameter of 14 nanometer. Creatine in different time in different time intervals 3 microliters of the sample was withdrawn and rapidly vitrified. The moisture vitrified in the ground is transferred to the cryo holder and subsequently introduced into the transmission electron microscopy. Without additives, time 1 minute. Shown on representative cryo-10 figura da sample vitrificada in 1 minute, showing that nanostructure from electronic denser region V, low dose of select area electron diffraction, shown that the nanostructure are composed of amorphous calcium carbonate. Using stain cryo, we can observe that the sample of what is a process of evolution. Here, we found the coexistence of 2 morphologies, as shown in Figure A. Figure A, the size distribution de la nanopartícula is shown in the Figure B. In Figure B, with its representative log normal fit, showing a main diameter of 5 nanometer. In the figure also we show nanorod with lateral dimension of 5 nanometer, but log axial dimension of 13 nanometer. Here we can see that the nanoparticles are like building blocks stacking of the 4 nanorod. The nanorod length distribution is shown in image C with its respective log normal fit. Data was collected from several sting images. sampled in 3 minutes. And in the figures A, shown on representative image of cryostem of the sample vitrified at 3 minutes. The size distribution of the dense nanoparticular wing spheroidal line morphology shown in main diameter of 20 nanometer. We emphasize what this type of particular morphology govern this sampling. Figura 9 shown a characteristic electron diffraction pattern obtained from the spherical nanoparticle of the figure 9A. The size indicates the presence of several crystalline dominions. However, it still shows the diffuse ring characteristic of vitreous eyes. The integral angular intensity of the diffraction pattern, figure 9C, for the sample of vitreous eye shown broad peak at background due to the eyes. However, the sample shows the presence of peak that fit the battery phase pattern. Without a sample time 7 minutes, we form dense nanoparticle with spheroidal. However, we observed increase in the main diameter of 25 nanometer. Show the side adhere from the facet structure, we show the formation of the calcite structure. However, this is a strong signal of the vitreous eyes at the bottom of shown is the rotational base with only reflection 120 is observed. Ainda no mês de 7 minutes, na mostra, showing in the indirect formation the calcita phase through the dissolution of nanoparticles from the baterita phase, the dissolution atom for 2 rhomboedra of the calcita phase, shown a zoom in on the region where the facet nanostructure are born. This nanostructure is analyzed by EDS mapping in G. The element map of calcium, oxygen, chloro, and sodium are shown in EDS spectrum, and the atomic percentage of calcium y carbon correspondiente of the regions marked por white the dashed lines. Okay. Wing additive, 3 minutes. We can observe that sample was is dense liquid state. In G1 minute, we thought nothing. In G6 minutes, we can observe denser center of the material of the sample does not have a particular. The sample does not have a particular texture. The box market represent the areas of the ears, showing the presence of the calcium esgue. In aim unit, we can observe an evolution, the sample present a nanoparticular texture with a main diameter of 10 nanometer. However, sites most showing that nanoparticles are amorphous. Analysis do EDS, mapping, elementos mapping of calcium, oxygenio. Conclusion, direct observation of the crystalline phase batterized in nanoparticle of 20 nanometer, direct observation of the dissolution mechanics recrystallization from a spherical micrometric nanostructure of the batterized phase to rhomboedral particle of the calcium phase. When additive, as particle acid reduce the availability of calcium or carbon ions, the amorphous calcium carbonate needs for its transformation in crystalline calcium carbonate. Bom, agradecimento. Thank you.
Thank you, Raquel. We are on a tight schedule, so questions can be made during coffee break that comes after Carolina's presentation. And our final conference, our guest has a flight to catch, so we have to keep on time. Thank you. And Carolina is one of the main organizers that made this meeting happen. And she also is a postdoc in our lab, and she's a teacher at the Federal University at the Polo of Duque de Caxias on cell biology.
So, good afternoon, and many of you already know me. And I will talk about a little bit about the work that I did in Munich when I was in Martinsried in the beginning of this year. And before we talk about the Paratrypanosoma confusa, that has a very confusing name, and I would like to talk a little bit about the trypanosomatids. So the trypanosomatids are these protozoa, uniflagellate protozoa that are parasites. All of them are parasites, so they can they need one host to complete their cycle. They can have just one host, and we call them monoxenic parasites. And but they can have more than one host to complete. They need more than one host to complete the cycle, and they call them heteroxenic parasites. And as you can see here, we have some important representatives of this family that are the ones that causes diseases in humans. So we have here an epimastigote form of ticruzi that causes the Chagas disease. We have here promastigote of leishmania that causes leishmaniasis, and here trypomastigote of Trypanosoma brucei that causes sleeping sickness in Africa. So, and here we have Strigomonas culicis, I think, I can remember, and that is a monoxenic parasite, so it is a parasite just for the insect that they parasite. And as you can see, these cells, these trypanosomatids, they can have many types of shapes. So they have several morphotypes, and they can change these shapes during their life cycle. So if they can change the shape, if they change the host. So they have several interesting structures. And here I brought some of them for you to understand the next slides of the Paratrypanosoma. And here, for example, they have only one flagellum. They have the normal 90+2 axoneme, but some of them have this cytoskeletal structure associated to the axoneme that's called the paraflagellar rod. So here we have a quick physical etching made by Professor Thais Souto Padrão, Vanderlei de Souza, and John. And you can see here the axoneme, and below you can see the fibers that made this paraflagellar rod. And these cells also have a cage of microtubules that are positioned below the plasma membrane. They protect the cells, and they are responsible for these different morphotypes. So you can see here a layer of microtubules below the membrane, and here a more recent work of our group where we show the arrangement of these microtubules and how they are positioned, and it starts during the cell morphogenesis. Another interesting structure is the flagellar pocket. That is invagination of the plasma membrane from where the flagellum emerges. The thing is that this region of the membrane is the only region of the whole cell body where the subpellicular microtubules are not present. So this is the only and restricted site for all endo and exocytosis in this parasite. So the intracellular traffic is always has the main point is the flagellar pocket. So here we have a representation of the flagellar pocket of T. cruzi, Brucei, T. cruzi, and Leishmania. And they are quite similar, but they have some differences in composition and also in morphology. And if we can see here in this scheme, that the flagellar pocket has some cytoskeleton structures that are also different between these three important medical parasites. So in T. cruzi, the cells have another important structure, that is the cytostome cytopharynx. Some trypanosomatids can have a cytostome cytopharynx, that is an opening on the cell membrane that invaginates deeply into the cell. And in T. cruzi, it is the only site for endocytosis. So all endocytosis in T. cruzi happens through the cytostome cytopharynx, and the flagellar pocket is only responsible for the exocytosis in this model. So this is the model that I actually work. So all my work is focusing on the characterization on the endocytic pathway of T. cruzi. And here in the last years, I was like trying to describe the autostructure of the cytosome cytopharynx, how it is organized, how the cargo enters through the cytosome and leaves the cytosome and reach the final organelles of these cells that are the reservosomes. So this is a tomography, normal tomography of blast-embedded samples, where we can see the region between the flagellar pocket and the cytostome, the opening here, and the cytopharynx going deeply into the cell, accompanied by 7 microtubules. So keep this in mind, because we will need this for the paratrypanosoma. And some of these trypanosomatids may also have a contractile vacuole. And as Kildare already mentioned it, I won't speak too much about that. But this structure is related to the osmoregulation of the parasites. And another interesting structure in trypanosomatids are the kinetoplasts. So the kinetoplast is the kDNA, is the DNA of the mitochondria that is all packed together in a special region of the mitochondria that we call the kinetoplast. So in different trypanosomatids, this kinetoplast can arrange, this DNA can arrange in different manners. So you can see here, this is T. cruzi, but you have here T. cruzi, but another morphotype that is a trypomastogote. So During the differentiation, they can also change the topology of the kDNA. So this is another interesting structure in the trypanosomatids. Well, what about the paratrypanosoma? Where is the paratrypanosoma in all this? So the paratrypanosoma confusum was first described in 2013 by the group of Julius Lux. So he isolated these cells from the gut of Culex mosquitoes. And they analyzed it morphologically. So he found one single cell, a flagellated cell with a kinetoplast. It has also a normal flagellum. And he described it very superficially, the morphology of the cell. But the most important of this work was the phylogeny that they made with the other trypanosomatids. We put the paratrypanosoma in the root of the trypanosomatid family. So this is a very important point because in the kinetoplastid we will have free-living parasites and we will have parasitic parasites. So paratrypanosoma was put in between these two major groups. So these cells may give us some hints of how it was the evolution of the parasitism in this, for the trypanosomatid family. So they also made some other morphological descriptions. So they describe that the cells in culture, they have many morphotypes, so they can be aptomonads that are small cells with a small cell body, and the flagellum attaches to the substrate and spread, and they can form this agglomerate of cells. And they also described a promastigote form, where this promastigote form may have cytostome cytopharynx. So these called my attention. And then we started to work with the paratrypanosoma, me, Marcia, and the group of Professor Cristina Mota. And then Vanderlei once, in the beginning of February, asked me if I wanted to go to Munich to train cryo-EM, how to do cryo-lamella and cryo-ET. And I said yes, and he said this to me on a Monday, and I travel on a Friday. And so I packed up my things and went to Martins Reed to learn how to do cryo-EM, cryo-lamella. And of course, I couldn't take my actual model, that is sticker's eye, so I took another parasite that was important for my work, but it's not pathogenic for humans, so that's why I took paratrypanosoma with me. So Matthias already gave a spoiler yesterday about what we did, and this is now summarizing all these steps. So we plunge frozen the cells. It was like he said, we took like 5 microliters of concentrated cells. We put on a grid. We plunge frozen these cells, clip them, and put on the cryofib, make some lamellas, and then we went to the cryos to make the cryo-ET. So this is the grid, the plunge frozen grid under the cryofib. We can see the cells here, small bubbles. Can you see? It's a small bubbles. They are all packed together in the center of the square of the grid. And then we selected some regions to make the lamella. And then we mill the lamella in these regions. And you can see here the face of this lamella. There are some cells here. And we saw that we have material there. And then in this first attempt, we made 3 lamellas, and that was really good. We could use all of them. And this is the pictures of the lamellas in the cryos. So this is lamella 1. You can see here some features like the kinetoplast is very easy to see. Here we have the nucleus, the flagellum. And in lamella 2, we also have very impressive structures. It was really, really easy to see, to identify the region of the flagella pocket, the nucleus, the kinetoplast. And then we started to pick up regions, important regions, the regions that I was interested, that was most of them in the anterior part of the parasite, that is where the cytosoma cytopharynx should be. So we made many tomograms. I think we made more than 50, right? Tomograms during my stay there, something like that. It was a lot of them. And in this first attempt, we have like tomograms with a good quality of vitrification. Some of them not so good. You can see that there is bad vitrification between the buffer between the cells, and some of those are very bad with the vitrification. But even these very bad samples, we had like a good result. So you can see here, I can't see, sorry. And you can see here the nucleus, the glycosome that are structure related with the sugar metabolism, and the acidocalcisomes that Kildare and Ingrid already told about. And we can see here that we can clearly see the nuclear pore of the cell. So here is sequenced Z of this tomogram, and here inside the glycosomes we can see some fibers. I don't know if you can see, but I can see some fibers here inside these glycosomes. That was really interesting because we never saw these in the other models. And this is another tomogram of the anterior region. So here we have the flagella pocket, the flagellum, and the first thing that called our attention was these fibers inside the flagella pocket. These are not something that we used to see in other trypanosomatids, so this called our attention. And also the region of the contractile vacuole, we could see some densities at the membrane, like a coat that makes like waves. So in this other movie here, I don't know if you can see, but we can see some waves on the membrane of the contractile vacuole that are those that Matthias showed you yesterday. So he made some subtomal averaging, and we are working on that. But it reminds me a lot of the retromer complex. So it's a very particular complex. It has like any structure well established, and this reminds me a lot of that. So we have here another tomograms. We can see the kDNA. So this is the DNA, the mitochondrial DNA. Here the region of the flagella pocket, the flagellum, and the region between these two structures. So Vanderlei asked me if I had seen a very common structure in trypanosomatids, that is the tripartite complex that keeps the kinetoplast associated with the flagella pocket. So this is very common in this parasite. They have this association. So I couldn't see, but later a paper was published, and I said, oh, maybe I saw and I didn't know, and maybe it is there. I just don't see with my eyes. And this is another one when we can see the subpellicular microtubules. We can define each one of the protofilaments in this tomogram, and also the mitochondria and all the crista and some densities in the membranes of this crista. And one of the most astonishing tomograms that we had was this one where we can see I finally found the cytopharynx. So I tried to make a scheme of the parasite. I'm sorry, I'm very bad in drawing. And you can see here the flagellum, the cell body, and there is a small invagination here that comes from inside the flagella pocket. And here in green, we have the kinetoplast in blue and the nucleus. And this section here, the somogram, was like if we made a cut like this, right? Like this, sorry. So we can see part of this invagination. And as you can see, we still see inside the invagination those like filaments, those repeated structures that we have no idea what they could be. And many multivesicular bodies very close to this invagination, microtubulus running along the invagination, as well as we already saw in the Trypanosoma cruzi. So I strongly believe that this is the cytopharynx of the Paratrypanosoma, and is completely different, not completely, but it's kind of different from T. cruzi, because in T. cruzi it opens from outside the flagella pocket, and in Paratrypanosoma it opens from inside the flagella pocket. And we can see here coated vesicles surrounded by the ER as well. We can see the mitochondria and the kinetoplast. And I tried to try to understand what were these things inside the cytopharynx. So we have shapes like a square, several small points, like a string of, how do I say that? Strings of bolinhas. Peas, exactly, this is. And we are trying to figure out what is this, but Marcia made some SEM showing these cells. So you can see here there is some cells that has this coat outside. You can see here better. So we don't know if this is like a carbohydrate, glycoproteins that are on the surface of these cells. You can see here in higher magnification. This is the model that I did from this tomogram. And this tomogram you already saw yesterday with Matthias. So this is the region of the flagella pocket. Again, a lot of content inside this flagella pocket. What we called our attention here is these filaments in the region of the entrance of the flagella pocket here, and also some filaments around the neck here in this region, around the neck of the flagella pocket. So this is very interesting because I never saw these in T. cruzi with plastic embedded samples. So maybe they are being lost and we didn't catch it, but it may also be new proteins or new constructs in this parasite. Okay, I'm finishing, Marcia. And okay, so here just for you to see that I pick up two different types of filament at the entrance of the flagella pocket. So here we have like a helical shape filament. Here is a more straight filament. So there are different types of filaments in this region. And also, and I wonder if this is a clattering lattice. Maybe John can say if this is a clattering lattice. So we can see here in the membrane of the flagella pocket. So this is the region where the pocket opens, start to open, and we can see a lattice of clattering here as well. And again, these coats in the surface of the contractile vacuole, this small wave-like, and another type of coat here, and in this vesicle as well that are close to this structure. So this may give a hint of another type of intracellular traffic that also plays function in the contractile vacuole. And this is a lamella that I did. I was very proud of it because it was below 100 nanometers. So we had little volume from this tomogram, but we can see many, many things. So again, the nuclear pore complex, the mitochondria with the many densities on the surface of the membrane. And also here we can see, oops. Where is the movie? Why the movie is not playing? Oh, sorry. Yeah. So you can see here at the anterior region of the parasite, a lot of microtubules and some filaments around this vesicle here that I don't know, I have no idea what it is. And the flagella pocket, the flagellum, and this incredible membrane system that this parasite have. It called my attention. And here is, I tried to make a montage, so I took a tomogram here, and a tomogram continues with this area. So you can see the superlicular microtubules, and they are like, they start to disappear when it enters the region of the flagella pocket. So they are like, they become incomplete as it approaches the entrance of the flagella pocket. And in this region, we can see like it was connections, junctions between the flagella membrane and the cell body membrane, like it was like desmosomes. I don't know, maybe it reminds me desmosomes. And here, you can see here the microtubules are disappearing, coats on the membrane of this structure, and here the filaments. And this is our work in progress. We have many things to do. We have many data to work on, and that is a chapter for the next chapter of this work. So I would like to thank Vanderlei for sending me to Martins Reed and to my endocytic group. There is one missing here. Pedro is not in this picture. He is my undergrad student. And people from Martins Reed, Jorgen and Wolfgang for receiving me, and especially for Matthias that had a lot of patience with me to teach me during 2 months in Munich. So thank you all. Thank you to Senabio. Without Senabio, we couldn't have done all the work that I did during my career, and now, especially now, we can do many of, with the new microscope, many things on cryo-EM and the financial agency FAPED. Thank you.
Thank you, Carol. We're having a coffee break, and please be here on time because our next speaker, Dr. Marin Van Hill. Okay. We have to start sharp.
Colleen, I want to make just one public statement.
Oh, sure.
You and I have not been able to talk about this. You have made a major discovery that no one knows about before. There are 2 types of contractile vacuum in the lower animal kingdom. There are ones that are permanent, just open and close, and there are ones that completely merge with the membrane and have to be recreated each generation from platinum-coated plastic. And you've just proved that this organism has that second type. No one ever knew that. Dictyostelium is the other example, which that is so well studied. So congratulations.
close the door. Please, we are going to start. Fernando. Encontrou o local, já está definido? É aquele mesmo?
Proponemos esto, Vanderlei. En Leme no quieren hacer reserva.
Sim, é quem chegar.
Quem chegar, porque podemos chegar cedo,
Mas
6h30.
eu diria o seguinte, acabou daqui, vamos direto.
Não, 6h30.
Não, sabe por quê? Porque se as pessoas saírem para ir para o hotel, para
Quantos
tomar
são?
banho, é mais ou menos aquilo, 14, 15 pessoas. Okay, we need to start. That is the last conference of this symposium, and it's a pleasure for me to make a very brief introduction to about Mary Van Hill, because he has a problem to travel and he cannot lose the flight. It need to be at the airport around. At 7, okay. So that's, we are going to start. Well, Mary Van Hill, I would say that he's a Brazilian, although he was born in Switzerland, right? Well, but he came to Brazil very young and stayed for several years here. So he speaks Portuguese very well. And of course, I know him by his contribution to the electron microscopy, but I have never met Mary. And during an international congress of microscopy that took place in Durban, in South Africa, I don't remember the year, but we were all in the invited people in the same hotel, huh? For 2000, my God, the time goes on. 2000, for me, it was a few years ago. But then, I was with Marlene in the bus talking Portuguese. Then he came to me and said, well, do you speak Portuguese? Are you Brazilians or from Portugal? We are from Brazil. I start to speak Portuguese. And I said, and why you are not in Brazil? So, say, let's do everything for you to come to work in Brazil. And he did this. First, came give courses every year, several courses, on cryo, single particles, and things like that. And that is the origin of the L nano, at least the microscopy portions of the L nano in Celepen in São Paulo. Indeed, the first cryo microscopy center in Brazil. That is still going very well, where people work, and you saw several presentations during this symposium. He also creates an important center in the Imperial College in London. He was director of this institute at one of the most prestigious scientific institutions of UK. Indeed, I visited Imperial College 3 weeks ago, and it's really now with new buildings, and is growing. And also worked in Leiden, at the University of Leiden, for several years, and now he is shuttled between I don't know where, and Campinas. I'd say that Mary is a kind of, it's not only an electron microscopy, it's a theoretical physics, very specialized in light optics, so with many ideas, and I'm sure that his lecture will allow him to show is how he see this cryo change in foundations of physics. So that's we are all look for, and I will tell you when you have 5 minutes, so that we can allow you to take your plane.
Thank you very much for inviting me here. It's always a pleasure. And you are the person who first invited me back to here. I must say I met my Brazilian wife, current wife, in London and not in Brazil, but we're very happy to be here at the moment. So yeah, so the first time I came to Brazil was a little while ago, and before, yeah, well, actually before I'm sorry, my talk is going to be a little bit different from normal. Yes, I have formulas in my talks, so I have to warn you. It's going to be a little bit of this stuff, but I'll try my best to explain here what everything means, and things are not as difficult as you think they are really. There's maybe something I recognize immediately here. No. No. Well, there's uncertainty principle here. Oops. I talk about it maybe at the last slide. Delta. Oh, that's the wrong slide. Oh, my. Oh, let me just use this one here. A happy face? Where's the... here. So here's the Heisenberg uncertainty principle. That comes in at the very last part of the story. And anyway, what did you see? There are women in the story, see? And one woman is worth 48,000. Oh, okay, no, let's not. Okay, so anyway, let's just go into the science. And to the science where this is, here. So the title is a bit different from usual. The talk is also a bit different from usual because I really go back to some basic physics. But before, I first have to still travel to Brazil. I'm way in Europe and at the remnants of the Second World War. And that's my first lesson in statistics almost here. You see here, this is I'm a bit nervous. So here, oops, that's the wrong one. So this is a Heinkel 101, and this one didn't survive very long. I think it had like 12 flight hours before it crashed out of itself. And my father was a resistance student, and he didn't accept the German invasion of Holland. So they caught him, unfortunately, and put him in a concentration camp in Berlin, and where he was forced to build these Heinkel 111 bombers. And that was one of my first lessons in statistics. Because my father said, all you could do without being executed, oops, it's not reacting as fast as I thought, is to play around with the statistical tolerances. And that would make then the planes be less reliable, and they couldn't pinpoint it to one individual because it's the statistical action of many. And so that's what... But luckily enough, there was an end to that misery. He survived 4 years of that, and immediately after the war, my parents got married in Holland. And when he finished his degree, we took the boat to South America, and that was in December 1949. And with the modern microscopes, you can see here on the back, there's a little boy there. And he's arriving in Rio de Janeiro, actually just a few hundred yards from here, at the old port and at the Central do Brasil. I couldn't find the picture because it was not online. But so that's it. But I mean, you do recognize this here, yes? In Rio de Janeiro, in 1949, the view here to Barra da Tijuca, and. We'll go back there later. This is a dangerous place nowadays, apparently. But this is the reason I grew up in Brazil and went to school in Brazil, and I was forced to to make this walk 7 times or so because my mother needed to send pictures to the grandparents, yes. So, and my father was the director of that milk plant from Nestle in the background here. And you see him here with Pieter van der Broek, who was from Holambra, the Dutch colony here. And he was the gerente, the director of the farm. The farm was a beautiful place. I'll get back to that. But anyway, here, a very important thing here for my career is just around the corner here. Yeah? Here, back there. You can look here and look to the left. The other side of the street, at least when the train passes by, there was a Maria Fumaça. And this was the Cerâmica Porto Ferreira, famous factory here in Brazil. Unfortunately, it was just sold out of the family. Dr. Nicolau passed very recently, but I spoke to him until his 90s. But I'm not talking about him right now. I'm talking about his son, Nicolauzinho. Nicolauzinho was a few years older than I was. And he got a present for his birthday or Christmas, I don't exactly remember. But and that was an optical kit of this nature here. This is a modern version. You could make microscopes and telescopes. And he told me, since I was a few years younger, he said, "Não toca, não. You're not allowed to play with this. It's mine." So yeah, well, so I had to go to my mother's desk and get the magnifying glass to do my first experiments in Fourier optics. Here you have a Fourier optics. You have a plane wave coming from the sun, and that would focus at a single point, which structural biologists will call a delta function, or mathematicians will call a delta function. And they also cared for the theoretical aspect. This was the practical exercises. And here the theoretical exercise, I just needed to grab the book from the library. This was a book that my father got when he was a teenager. Well, a bit later. He was born in 1990, so yeah, well, teenager when he got that from his parents. And it's a beautiful book, fantastic book with fantastic illustrations and wave patterns and I've used many of those pictures in my lectures at various universities. This is the Dutch version. I have an English version that I bought secondhand on, so I really can recommend that, to search for that book in Amazon or whatever you can. But okay, so this is a bit the scientific side of Brazil. Of course, you don't go to Brazil for science, you go to Brazil for Carnival. And this is our little city, Porto Ferreira, here in 1954, and it was very, very busy. And a year later, the Carnival was at home. And why was it at home? You see me here picking up particles, confetti from the floor. And so this was the pandemic. And my mother was supposed to be a medic, but because of the Second World War, she wasn't able to finish her studies. And during the pandemic, of course, you couldn't have contact with anybody else, so my parents organized a secure environment where you couldn't get infected and be condemned to spend the rest of your life in an iron lung. But of course, they did the indoctrination for my sister and I, for example, into vaccine research. And this little rat here, or mouse in English, was being here immunized or just creating antibodies against the polio. So this was one, you just give the right books to the children and then everything. But anyway, we didn't spend very much time here because my father then didn't want to continue at Nestle at one point. So here is between 1949 and so last year he worked for Fokker here in Galeão, and we used to play on the beach here, so you actually can recognize these mountains tracks here. It's exactly the same, but it became more and more dangerous. Yes, so here's my sister and I enjoying ourselves, and here somehow it generates violence attitude here. And well, as you all unfortunately know, that in the other side of town here, the same hotel, the Windsor Hotel in Barra, was the site of an assassination of the members of a medical meeting there that happened across the street. So 4 people were just, well, it was a change of, well, anyway, Brazilians know the story. I won't. It was violence, random violence, and on the wrong people. But anyway, so it has, of course, nothing to do with us playing there on the beach. Just, I am not that aggressive, just for the photo. And, well, not all. Okay, so, and then, so then we went back to Europe. You see the boat is now going from the left to the right. That was in January 1960 that we left Brazil again. And so then I went through a normal career in physics and a secondary school, and I landed at doing theoretical optics at the University of Groningen. So what I did there was actually something that I had almost forgotten, because and during the pandemic I got back to basics, so to say, and so what I did there was an experiment, which was the double slit experiment. Here, this famous Louis de Broglie, he got a Nobel Prize for his PhD thesis, and he postulated that particles like electrons have a wavelength, and very friendly. You can nowadays just search the web and listen to the stories when he's interviewed. Those things are. But I mean, so the idea is that you have electrons coming from an electron gun up here, and then you let them pass through 2 slits, and you send them off here, and you collect the arriving electrons. And this is really a rain of electrons. This is really quantum statistics, and slowly this builds up. This is from a group in Japan, this image. In those days, in the when did we do this one? In the early 70s, 50 years ago, there were no cameras where you could really film, so everything was just on photographic film, and you integrated on photographic film. But we did very interesting experiments here. It was, I didn't realize how interesting they were 50 years ago, otherwise we would have to. So here is actually a hemocyanin molecule, one molecule, and these lines are the interference patterns from the double slit experiment, and this one came from a theoretical physics group, theoretical optics group, and then from this image you can find both the amplitudes and the phases of the wave, and do these things. It's a bit more complicated than that, in the sense that the equipment was what we call a biprism, and so you have to take an electron microscope and change it, and then put a little hair in there that splits, that you can charge, and the virtual sources come from 2 different points, and they interfere in a certain area, and that allows you then to do things that you couldn't do. What became completely clear to me at the time, I came from a group where everything was analytical functions and theoretical mathematics, and what it actually was in reality is, as you see here, is counting arriving electrons, and that counting of arriving electrons was not so clear to people in those days. So, okay, then we go to the phase of cryo-EM, and I'll just go through the 3 most important slides, were most recent the developments, and that one of the most important things is the arrival of this kind of machine, and especially where you can control the microscope. You don't see it anymore. You can control everything on a distance, but these cameras. These direct electron cameras, where you count each arriving electron, that really made a big difference, and that led to the so-called resolution revolution, as Werner Kuhlbrandt had published it. He just retired 3 weeks ago, and these new cameras allowed you, together with all the methodological, to look at proteins at high resolution, and you could see the side chains, and this is I took from a 2.2 angstrom paper. So that became then suddenly cryo-EM became the big thing, and it started to compete in terms of resolution with classical electron X-ray diffraction, and but you didn't have to grow crystals anymore. You could actually just do these individual things, and then came, of course, the recognition for things like that. And here, this is in 2017, the Wiley Prize for their pioneering of cryo-electron microscopy, and with Richard Henderson and Joachim Frank. And later that year came the Nobel Prize, where Jacques Dubochet was included, and he certainly he developed the freeze plunging techniques and made that into a routine operation. And I was at least invited to assist the formalities there, and that was a very nice experience. The instrumentation, there were a number of things that were very important for this to happen, and one of the main things was the instrumentation. While very bad things happened on one side of Berlin, on another side of Berlin, there was Ernst Ruska developing the first electron microscopes, the first microscopes that had a higher resolution than the optical microscope. And this here, his design here was from the 9th of March, what was it, 31? And he got the Nobel Prize in 85, I think it was, let me see, 86 here. By that time, I worked in that institute in Berlin, and he would come in still. He had the office on the other side of the corridor from me, and I had helped him with photocopying machines or something like that, but that's all. He was just doing the historical reviews of the development of the electron microscope, and I knocked on his door and asked if I could take the picture, and was okay. So, and then years later, I digitized those pictures, and only when I digitized those slides did I notice that he had cut himself here, sparadrapo here on his face, and so that was very nice. And I actually later got the Ernst Ruska Prize and had dinner with him, and he was. He built these these instruments were first high resolution instruments were built in an academic environment, yes, and then later now the industry kicked in, and Thermo Fisher of all they can do it much better than this was the first experiments. Here you had these towers. The outer tower was only to prevent the wind from hitting the inner tower, and from the inner tower so that you wouldn't shake. And the underground passing by wouldn't also cause vibrations. And the inner tower was suspended from the inner tower was a cage, this orange thing here, and there on a platform was this the instrumental. Today things are very different. I mean, you open the box from your krios, and you push against it, and it slowly goes back and slowly dampens out and is still again. So I mean, the technology to make that into a mass production device is but I mean, much of it happened here, and here is after he was retired by the time. Who hired me was Elmar Zeitler. And this is Fritz Semlin, who was one of the developers of the highest resolution microscopes, certainly in those in the 1980s. And people would then come from all over to use his microscope. This is the last day of the Suleika, and they were transferring the Dietrich lens, which is superconducting liquid helium lens, into a CM200 type of microscope. I was very happy to hear that from one of the colleagues here that FEI, I'm not sure that whether I'm allowed to even say it, but that they are now again picking up that liquid helium line and building things. So I'm not sure that I'm giving confidential, but I'm very happy to hear this, because this microscope, or this super liquid helium, was the device that I chose for. And one of the people that came there was Richard Henderson, who did 2D crystals from Bacteriorhodopsin, Halobacterium halobium, and as it was called in those days, and it was the best images in the world, and they were tilting, and they used the microscope. And until the present day, in the institute, there would be the guests and I in the middle of the night, because, or we would go for a pizza at the end of the day, because everybody else at 5 o'clock would close the door and go to by underground, go home. So we had a lot of chat, but this is my son who took this picture in New York when we had a pizza the last time. So specimen preparation, that was one of the main important things that developed, and that was Jacques Dubochet was the great hero here, and there were some earlier developments. I mean, in San Francisco, but he made it into a very practical way to rapidly freeze samples, and he loves water specimens embedded in water. And I took this picture here while I was canoeing with him 20 years ago. Wow. And so the technique, you've heard that 1,000 times this week of how to rapidly freeze plunge your samples. This is the first machine that we built in Berlin. We had a hard time to get good samples, and this is the 3 millimeter copper grid that you use to put your sample on. And then you freeze it rapidly, put it into a vitreous water state, and then this is the diagram that one of our ex-students drew, and here you have hemoglobins in this layer of vitreous water. The vitreous water has a density almost identical to that of water. It's not like a crystalline water that expands and then disrupt all the structures. And from these projections, you can then calculate the three-dimensional structure. The Jacques Dubochet made this into a routine technique, and this was here during. I took this picture of him in September. 2017, Delft Technical University, which was one of my alma maters, and he took the last bike, and I had to get a cab to go to the lecture. So, but I mean, at least I had this picture of him. So I kept something that is memorable, and so we also always had great fun together. And then the other thing is from these types of images, you get all these projection images, and how do you make that into a 3D structure? That was the next issue, and so that's I had an interaction with Joachim Frank about phase contrast transfer functions in partial coherent illumination. I'd written a paper about it, and I'd send it to him, and he said, "Yeah, you don't understand this," and that's when I understood that he didn't understand the theory behind these things. And so he had the paper on the envelope function, envelope for the partial coherence, and an envelope function only suppresses things, and the actual operation is a convolution-like operation in the backfocal plane. Anyway, let's not go into the details, but anyway, so I published that paper independently. And he never cited it, I think, but it actually refutes the partial coherence theories that he had published earlier. I didn't make a big point of it at the time. I had taken from our lab in Holland samples, from negative stain samples that we were looking at every day, and I had met him at a meeting in Basel in '77. Joachim Frank, and we thought it was a good idea to have a technique to find these different views of molecules in samples, so that you could understand things better, because you get all this mixture of things. And there were a couple of interesting things that I had looked at. One is we had some hemoglobins, worm hemoglobins, with something in the center and without something in the center, and that worked okay. So this we were aligning these things at the time, and then there was a big breakthrough when we started to use multivariate statistics to find these different classes and understanding data in an abstract way. This is all visual. You project everything on a low dimensional space, and you draw these things manually at the time. What happened is the following. Jean-Pierre Bertaudière was a mathematician who was hired at that lab. I had met Joachim Frank. I decided to go there for 6 weeks, and during those 6 weeks, we're using a computer called a PDP-11. Here you see Jean-Pierre Bertaudière in front of that. Here you see my little mess here in this corner. That was my desk. And so this computer during the day was used for the administration of the New York State Health Department. And so we were there in the middle of the night normally, and then go to the pub at 3 o'clock when we started to fall over. And and then we understood that this was very powerful for doing just that. And so that was an immediate success. And there were all kinds of further techniques that had to be developed here, like automatic classification, getting the angles, 3D reconstruction, surface rendering instead of cutting out, cutting out balsa wood sheets. And you needed resolution criteria, all these things. So these things I have done later independent, but and we had, Joachim and I had applied for a grant, and that came finally through in 2000, in 81, 82. But things were not going in the right direction, and so he was, when he was reporting these events here to the literature, I went to Marin and said, "Scrap the programs you are writing." He was a secondary player in this whole thing. And he changed this, so that didn't work. So we have, since 40 years, no longer been on speaking terms. And one of the things, for example, was that I had my Magic software system, and he had his Spider software system, and I had changed things enormously. And I had my paper ready in 1981. And we had agreed that it would go back to back in the same journal. And when I asked him about, I was ready 3 months before him. And, oh, did we agree on that? I have already submitted that 2 months ago. So there was things like that, and it was a complete, how do you call it, slowly level of irritation, which has made me actually go to Berlin instead of going there. So what was this? Okay, it was a headache. How is it going? Terrible. I have a splitting headache. I took an aspirin already. Yeah. So aspirin is a little bit. Spider is the software system that he had developed, and I used that for a joke. Somebody reminded me of that. That I had used a very similar one about 35 years at a meeting in Burggamen, and here at the meeting. Yes. So that was lovely to hear that. They remember that very, very well. So this is why it came back up. But so the decision was to go to Berlin and not to Albany, and so here we go again, and this is Ernst Ruska coming in, and the context with Fritz Semlin and liquid helium microscopy, and Elmar Zeitler was, I'm here honoring him here. He was very, very open to new ideas and let people do their work and research and so on most of the time. Like all humans, we have our ups and our downs, but it was really, this is a type of director that you wanted to have in the Max Planck Society, to be able to work, to have enough funds to go but I mean, I learned there, and for me was the idea was we need a liquid we need a liquid helium microscope, and so this is what I was looking for, and I ordered what was the first Polara prototype, and we had a meeting in Heidelberg, and with a few people, including Richard Henderson also, and a very important person in this story was Karel van der Mast. And Karel van der Mast was the head of development in Philips and then FEI at the time, and then he went into silicon production, a lot of more money there. I spoke to him relatively recent, but and I knew him from our first year in Delft Technical University, and he drew a painting on the blackboard, and I said, okay, I'll take it. So that was all the paperwork and the design that I've seen before, and just because I really trusted the guy. So yeah, we even spoke a few years ago still for the last time. But so yeah, if you buy a prototype like that, it's a prototype. And I had counted on that. We had a 200 kV microscope on the side to be able to always work. This microscope was, of course, a white elephant because once this started to work and work fine, the Polaras were sold like hotcakes. And then there was no manpower available to maintain this one and to update it because, I mean, this was the prototype of the Polara. Anyway, so it didn't have a long life, and I was still very sad when we had to dismantle it in 2013. I was already in Leiden then, and in Leiden there were the two fantastic Cryos microscopes, and they were, oh, this is Sasha Di Carlo, I should have said that. Sasha Di Carlo operating the microscope. It was a new world for me to have this type of microscope where you could count the electrons that came in. And here, fantastic new cameras. DQE, I'll come back to DQE. You pay a lot of money for DQE. But I mean, so it's a box, you don't touch it anymore, but sometimes you have to open the doors to make sure that there's actually a microscope here. It's not. And then what happened is, yes, at one point in a meeting in South Africa, there was this guy in front of me in the bus, and I was speaking Portuguese to him, and then he said, "Are you giving a lecture here?" Yes, and I saw them there sitting in the front row the next day, or 2 days later, I don't remember. And the next thing that happened, I got an invitation 6 months later to do an opening lecture of the SBBM in Caxambu, and it was lovely. And it was for me the first time that I came back to Brazil after 45 years, and I took the bus to Campinas. So Rodrigo showed that this morning. And I went back to Porto Ferreira, and one of my friends had a restaurant. He's now retired, but he invited all the people that were at my 9th birthday party to come to his restaurant. Well, it was fantastic. But going back to that Brazilian time here with my father, with Peter van der Broek, where he's looking at this is the model farm. This building is still there, actually, and here you see my sister and I on horsebacks, and this was the fazenda, the little fazenda for Mr. Peter van der Broek and his small children, and but then something happened. I was trying to find it back when I was a visiting professor in São Carlos, and I wanted to find this farm back, and it had now a different name. I couldn't find it back. And finally somebody told me it's now called Hotel Fazenda Gloria. And it had been changed, it had changed a bit. The Emir of Qatar had bought that farm. It was 600 hectares. And for each of his 5 wives, he had an apartment with 2 jacuzzis and 2 bedrooms. And for each of his 20 sons, there was also a separate building. The girls didn't count. They stayed in Qatar, I think. But he liked football, and so that's the reason why that little house was knocked down, and there was a palace of a hotel there. And that's when I found this while I was as a visiting professor in São Carlos. Rodrigo already told about that, and that was the first Brazil school that we then perpetuated until 2018. To 2020, we couldn't do it anymore because of the pandemic. But hopefully, we can still do it next year. And so the last Brazil school was 2018. And that was the beginning of Cryo-EM, so it's, the group gets bigger. This is in Mariporã. And in the meantime, I had a grant with, or at least a grant to visit here, and I helped also a little bit in the grant application. And so what was here the first Cryo-EM of Brazil was Crios of Brazil, was wheeled in 2018. We're now witnessing the second. And you see here that Rodrigo is having a hard time pulling in the new cryo mic, but he didn't do it all by himself. Luckily, he had some friends to help, yeah? Some strong friends. And now things like Mayaro virus structure determination is something that we have sharpened our teeth on, and that works fantastic. So all the procedures here, film, CTF finding, eigenvector analysis to classify the views, all these things. I shouldn't use the mouse, but should use this one here. So here the eigenvector analysis of these centered particles after CTF correction, particle picking. So you refine, and the structure of this thing is below 4 angstroms actually. The very important aspect here, I'm going to say a little bit because I'm going to stick around and talk about these metrics a bit later. So this is the metric that everybody now uses, the Fourier shell correlation. It's something that I proposed together with George Heraus in 1986. That's the first time it appears in the name appears in the literature. The citations are go all over the place for more political reasons than factual reasons. And what you do is you have two independent structures and you compare them to each other as function of spatial frequency. So out here in high resolution, you compare the whole ring here or in 2D images or the shell. And out here, you have a large number of voxels or pixels inside these rings or shells. What does that mean? That means that the correlations, because you have so many members, are pretty defined. The random noise component is very low. And therefore, you have a threshold curve, which is called the half-bit threshold curve, which drops off here and becomes, when you have a lot of close to the Nyquist frequency, you have something that becomes pretty flat. At low resolution, you have very few members of in a circle. And the statistics are very bad. And therefore, the threshold goes up to beyond 1 here. So this doesn't mean anything. This gives the impression that you know everything of the low resolution. Actually, it's the worst part in terms of how things are defined. And maybe I'll get back to that if I find the time. But the idea is this, yes, very important. And this is one of the main reasons why fixed threshold values of these metrics of Fourier shell and Fourier ring correlation cannot be correct. Because the statistics here, you have to adapt for the statistics. And the statistics, the number of rings, the number of sampling points in a small ring and the number of sampling points in a big ring is very different. And the statistics is more precise out here than here. So you cannot have threshold curves must always be dependent on the distance to the resolution dependent. So whenever you read actually something 0.143 is wrong, and just in the terms of not just wrong, but I'll get back to that later. Somebody looked over my shoulder when I was writing it, so I won't say anything more. But so instead of the so this is the Fourier shell correlation based actually this half-bit criterion is based on a definition of information by Shannon and his signal-to-noise ratio information, and that actually itself turned out to be incorrect, and so here's where we spread out to more general physics. So, and if you do it correctly, we now have the Fourier shell information that's published here in archives. If you have the things correct, you see that there is no information at low resolution. There is a curve still like the half-bit curve or the bit curve that's perfectly predictable. This is here hand-drawn, but nowadays it's actually already, it's a curve. And you see here that depending on how many particles you add up, the information goes up. And it doesn't really matter that whether the resolution goes up or not. Actually, you're integrating the total amount of information. That's what you're after. You want to have information. You have a lot more information on beta sheets and alpha helices here, whereas it doesn't really show up here very much. But again, the threshold curve is always a curve, and you need to have a correct metric to integrate that information. So here's the mathematics that's there, and here is the classical Fourier shell correlation. It has an oscillator that is between -1 and +1, and the Fourier shell information is defined from -infinity to +infinity. And this one plays a role here, but now not with the incorrect signal-to-noise ratio by Shannon, but that's now corrected. This is actually a patent applied mathematics at the moment. And this allows you, for example, to compare structures internally or between structures, like the spike protein here. And depending on how these are the same comparisons, but different thresholds. And you see the glycosylation level very different in both cases. And I will go to the next slide just to, I think, how do I do this? Click, click, click somewhere. Here? Here? Yes. You see here the spike protein, the S2 part in here. Sorry, I think the S2 part in here is the part that is most stable. You have the highest information density. That's the colors show that. And out here you have the lowest information density and most flexibility and glycosylations that are floating in the air. And let me go to the next slide. And this is exactly the same story, but then at a different threshold level. Again, you see here what one has to still scale these things. We have a project going on together with collaborators from São Paulo to look at the flexibilities and the resolution as function of place, and for all 1,000 spike proteins that have been deposited in the spike in the PDB structural database. Okay, now I have to run through the statistics here because I have 5 more minutes before I need to leave for get my plane. So here, this is classical. We're now going back to basics, and we have here Ernst Abbe and his Abbe diffraction limit, and how this looks like, what it looks like in a light microscope. It depends on the aperture of the microscope. This, by the way, is my mother's microscope from her study as medicine. I've last seen it from her brother in Wisconsin. Unfortunately, he passed away, and I have no idea where that microscope is now. But theoretically, this classical resolution stuff is given by the aperture, numerical aperture. And the way I like to really look at imaging systems in general is in this very abstract way. You have one Fourier transform system here, which takes the wave that comes out of the object here, and you Fourier transform that to the back focal plane. A second Fourier transform lens does the same, so that you have a one-to-one image from there to there. And of course, if you have different foci, it's the way how you organize it. The illumination system of an electron microscope is the same. It's not the way you are used to think of it in C2 and 3C and whatever, but there is a very clear, simple mathematical story behind this. And this is a linear system. What does that mean? Well, you have a Fourier transform here, object Fourier transform to here, another Fourier transform does this. And that means actually that you can predict things very precisely. So if you have an object and you use a linear imaging system, then you can predict, if you know the object and you know the optical transfer system, then you can predict what the image becomes. So here, this is the Fourier transform, at least the amplitudes of Marilyn Monroe here. You pass it through a low-pass filter, if that's what your optical system does, and you Fourier transform that back. And this is then, you can then mathematically describe this as here. The Fourier transform of the object times the transfer function is the Fourier transform of the output image. It's very simple. This is a linear system. What does a linear system mean? A linear system allows you to, if you have a specific given object and you have a given transfer function, then you can predict the output. That's what this is tricky, and vice versa. I'll get back to that later. This vice versa aspect here. It means that vice versa, it's pure mathematics. This is deterministic mathematics. That means that if you know the object, you know it deterministically, with infinite precision. And you know the output with infinite precision. You know the transfer function with infinite precision. And then everything just, this is deterministic mathematics. It goes both ways. It's very how do you call it? It's very unintuitive, but it allows you to do that. And Scherzer, Otto Scherzer used this idea also. Exactly here you have your backfocal plane, and here you can have like the spherical aberration constant of a function of the microscope, and again you can predict what the outcome is. Here is Otto Scherzer, and I took a picture of him at one of my first international conferences in 1978 in Toronto. And so what you have there is a test object, and you put it in the microscope, and that here comes the output image. The Fourier transform of this, multiply it by the CTF, and Fourier transform that back to that. And this is a linear optical system. What this means also, and that's a little bit tricky, because this is a deterministic mathematical function, you can go both ways. If you know whatever you know, if you know this with absolute precision, the image, if you have the properties of the imaging with absolute precision, you can calculate that one. And that's a bit weird. Because for a number of reasons that is weird here. Because so these are analytical functions, and the question is, are my functions analytical? No way. Here we have linear systems. This is again the image of Marilyn Monroe. I showed them, and it goes both ways, but both ways here is not realistic, because when you take an image of anything, you're collecting photons and electrons that are coming in. That means also that you can't go back. And why can't you go back? Oh, well, I'll come back to that later in a few minutes, because that's when I need to leave. But so there's still noise. So you actually have no idea what the object is. This function doesn't tell you how to calculate the only thing that you can do is, so the resolution, there are two different things. You have the instrumental resolution, that's the analytical function that we just looked at, and you have the results resolution. How much information did you harvest on that sample to be able to calculate, to have a good idea about the object, what the object is. And is signal-to-noise ratio a good information metric? Can I assess the quality of results? Not quite. Here we have Marilyn Monroe and noise, and we can add the two, so Marilyn Monroe on the rest, and then we see that it's very noisy. Now the signal-to-noise ratio on the right-hand side is here what's on the left-hand side. And you can start binning these images, and you can start binning further and binning further. So you have binned 8 by 8. One pixel in this image contains even 16. I overdid it. And 32. Let's go back to 8. But what you have here actually is in fact this. So you have this image and that image here, and this has a higher signal-to-noise ratio than that. Is signal-to-noise ratio information? Forget it. So is signal-to-noise information? No. So no way, to be very clear. And for example, if you have an electron microscope and you pay $1 million for a direct electron camera with a very high DQE, then what are you doing? Is the DQE of an image transducer a measure of its quality? Signal to the noise? No way. You're paying $1 million and you don't, for a metric, something that has never been defined. And the one of the nicest pictures that I've seen papers was by FEI, where they said they measured. I don't know if this was a follow-up to this paper from the group of Cambridge, where they stated they told these people that wrote this, well, we built the camera and we measured a DQE, and our DQE is actually 20% lower than you say it is. And so the manufacturer is telling the users that it's not as good as you think, and it has nothing to do with the camera quality of the camera. It just really has everything to do with the fact that the metric doesn't. The metric is not okay. And so there is now we have this paper, that archive paper. Okay. Now I have to almost decide politically whether we go on a bit. Here. Okay. So here is I'll take one more minute. So this is the linear transfer phase of an optical system, and when you do X-ray crystallography, you only have the diffraction pattern, And that means that the resolution ideas that you have here don't apply to systems like this. And is X-ray crystallography, where you're refining the measured amplitudes against the calculated amplitudes, is a nonlinear operation. The idea of a transfer function doesn't apply. And the R factor is actually a bad normalization here, because it's normalized only to the F observes and not to the F calls. So there's some hidden normalization in the program that will make this thing behave, because you can multiply a 3D map by a constant. It doesn't matter. It doesn't change anything. But the R factor changes. And the R factor changes. And so, okay, I'll stop with this slide. Just. I have a new metric. And this is the blunder metric, the blunder level metric. And so, and I didn't talk about the really thing that I want to talk about, so that I'll do that in Olinda, but so there's a lack of time and lack of correct flights. But I have the blunder. So what do we do? I've been talking about R factors. I didn't even touch upon the EM factors. But so if somebody shows that something is mathematically wrong, then we make that blunder factor here. We start off with 10 to the power 0, which is 1. If the person still continues to promote a false metric, in the second decade we have a 1 here. In the third decade, we have a 2 here. And so if people get, I mean, why am I doing this? Because a student starts to work on a blunder 10 level, I mean, it's most likely going to waste a lot of his time, yes? And so I would much rather see the new generation start looking at real things instead of this garbage from history, yeah? So, I said I would, but I will do 2 more slides. And so here is this formula. Signal to noise ratio is incorrect. It's a false formula published by Frank et al. Ali. And you can immediately cross correlation coefficients. You can have a negative value. Put any negative value. The signal to noise ratio will be negative, and that is outside of its definition range. So the formula is wrong. And it's been around there for, I mean, in terms of blunder factor, I don't even want to think about it. But and many people, and the problem is that this is used to justify criteria like 0.143 or whatever, any fixed threshold criteria. 0.5, there was a long paper, completely incoherent papers justifying this. So anyway, I already said this can only be positive, and this can be positive and negative. Just try a negative function value here. And I thank you for and then we go to electron microscopy. We'll keep that for Olinda. Thank you very much.
Any comments or questions? Very fast. And while he is organizing his
I hope I didn't bore you. That would be the greatest sin. Rodrigo, we have to run. Oh, one question.
Yes. When you showed that when you compare two types of diffraction patterns,
Yeah.
one inside the ring and the other dots inside the ring and below the ring until the radius is 0 from the center of diffraction pattern.
Yes.
Was talking too fast. What has the most higher resolution? I thought you mentioned that you have dots from 0 distance from the center of diffraction field, the radius.
Yes.
Higher resolution than if you have all the points in the maximum area.
Yeah, well, you see that if you go out, you get more information per ring, and when you get very out, it drops again. But it starts at 0. So it's like in X-ray crystallography, the zero order people you filter out and you don't know anything about it. You have to normalize that later back into the data.
Okay.
So sorry.
Where is the mic? Okay, I think that we are near the end of this symposium. Of course, we have to say thank you for all people that comes here from different parts of the country, from the different institutions of our universities and all the universities in Rio, and especially our colleagues that came from the different countries to give excellent lectures that, of course, was very useful for us. I would like to ask the director of Senabio to say a few words to finish this symposium and say that we have the coffee now at the same place. Okay?
Well, colleagues and friends, all good things in life ends in some moment. Our life ends. This symposium is ending now. But I am very happy because I know that this symposium was fantastic, unforgettable, and profitable. I would like to see you next year in the Cryo-M course and probably during the inauguration of another equipment at Senabio. Thank you very much for coming.
Okay. A special thanks for the organizers. My role was only to coordinate, but Kilda Miranda, Carol Cantor, Marcia Tias, that I don't know where she is. She's organized the coffee. But all people that collaborators of the cell structure lab and the Senabio, the microscopy unit, and some people from the macromolecular unit that work together to help the organization of this meeting. See you in, most of you will be in Olinda. That starts next Monday, or no, Tuesday. Tuesday afternoon. But then you have time to go to the beach, yeah, okay? So that's to, acho que some people are arranged to our invited guests for tomorrow to visit some places in the city, if you have time. And then some will be back tomorrow. John is going tomorrow, right? Who more is going tomorrow? Only you, Jose, yes, okay. But the others will be in Olinda. Gilder wants to say something? To show some jiboia?
Just wanted to thank the staff from Senabio. They worked really hard to put us through the whole nightmare with the construction and installation. So I think that without them, I said that in the beginning, but I would like to thank them again. It was really, really important to have this staff. They're really exceptional.
I forgot to mention the support agencies. I think it's very important to recognize the people that give the money to organize this symposium, so that FINEP in some way contributing to the Brazilian Society of Microscopy, in some way indirectly helped the organization of this Congress. FAPERG, that's also used money to support, and the companies, especially Thermo Fisher, that was one important support of this program of this symposium since the beginning, and then Carl Zeiss and Arpia that represents Leica, right? That also give you some support. That I would like to thank to all of them, and I hope that you will continue to contribute with the next symposium next year. All right.
One more thing, just to say that our doors are open, and we hope to see you back. So I'll really keep in contact with you so that we can continue our collaboration. We already have some grants going on. Vanderlei has some things already with Spain. I also have grants for them. So it would be really grateful to have you back here.
I hope that Bruno will come very soon, but not only for 1 week or 2 weeks, but for the whole life. That is what we expect from you.